let's get started, if the Panopto will ever start. All right, we're good. We are still talking about Chapter 15, genetics, all of its genetics. I don't remember the exact title for this chapter. Gonna go over a little bit of pedigrees still, because it seemed like the clicker question at the start of class with the pedigree tripped a lot of people up. Admittedly, it tripped me up at first too. I was like, yeah, I'm pretty sure it's recessive. And then I was like, oh no, it's asking about the parent, not the... So we'll look at that and see how we would get that right, in case anybody was still confused on that, because that is a very test-type question right there, that clicker question. Most of his are. Same old drill with the roll. 8:30 on the front, 9:30 on the back, name only. Please try and get yourself in the right section. I say it every time, never happens anyway. Here's a meme, I hope you enjoy. Oh, my other one didn't load. There should have been 2. I'll give you guys 3 next time, how about that? Just as an announcement, we don't have class on Friday, so don't show up to class on Friday. For those of you that aren't freshmen, we aren't doing the like Friday on Thursday thing anymore. That's not a thing. For those of you that are freshmen, we used to just hold Friday classes on Thursday, and it was a nightmare. Biology fact for you: 10% of all human beings ever born are alive right now. So let that sink in. Because we also haven't been around for that long, but that's pretty crazy. 1 in 10 humans ever are alive right now. Okay, the study tip, it's a little bit of an obvious one, but it's to eliminate distractions, especially cell phones. I see this happen to myself all the time, that I'm studying and then I start getting texts, or I want to look at Facebook, Instagram, Snapchat. I want to watch Netflix and study at the same time, because I can have The Walking Dead on in the background and study it. You cannot. I tried. Trust me, you can't. Okay. Now, I'm not saying that you need to shut yourself off in a room and power down every electronic device and hide away from the world for 3 hours, but maybe especially on an important study session right before a test, or if you're working on a paper or something, even just devoting 1 hour of solid studying, distraction-free, you will get 3 times as much done as if you had your phone next to you, right? Or if you had a TV show on in the background, okay? So eliminate distractions. All right, so this was pedigree that I showed on Monday. It's for hemophilia. Does anybody remember whether this was a dominant or recessive trait? Recessive. It's recessive, okay. How do you know? I heard a couple people say because the parents don't have it, but then one of the children does, right? Is it going to let me draw today? Oh, yes. So the parents are unaffected, right? Again, I will reiterate, filled in and empty does not mean dominant and recessive. People do that all the time. They think filled in means dominant, right? No, filled in just means they're displaying the trait. So Leopold has hemophilia, but his parents didn't. So the only way for parents to be unaffected yet have an affected child is for it to be a recessive trait, right? Now that we've talked about sex-linked traits, and someone pointed out last time that this would be a sex-linked trait, this is an X-linked recessive trait. What does that mean? I heard you mumble it. Carried on the X chromosome. So looking at this pedigree, how would we know that? Perfect. Always look for patterns. Do you notice that only males are carriers here? Okay. Again, with X-linked traits, because it's an X-linked recessive, both X chromosomes would have to have it That's a far lower chance, right? But in males, we only have one X chromosome, so it's twice as likely, right? And actually, it's more than twice as likely. But so look for patterns. Look for parents with unaffected parents with affected children, and then look for, if it's talking about whether it's sex-linked or not, look for the ratio of males to females. If it's like 90 to 100% males, you can pretty much say that that is an X-linked trait. Okay, now we kind of started on this, didn't get a chance to finish it, and I think it would be beneficial because of what I saw with the clicker question and some people not, well, a decent portion of people not getting that right. So first of all, what kind of trait? Recessive, dominant? Ooh, I heard a little bit of both. It's recessive. How do you know that? Yeah. Unaffected parents having an affected child. Now, people usually, where a lot of people make the mistake is they go, well, affected parent, so it's not recessive. No, keep going. You'll find the unaffected parents having affected children, right? Look at everything before you just jump to conclusions. So let's break it down. What genotype is that woman going to have? Somebody said heterozygous. Not. Homozygous. Why?
The reason why is because dominant would prevent it from actually occurring. So like number 4 and number 5 is probably heterozygous. And while the recessive is just completely recessive, it can't overcome the dominant gene.
Yeah, in simple terms, it's a recessive trait. They have to be homozygous recessive. There's not another option. There's no way for that circle to be filled in unless it is little a, right? I think sometimes we overcomplicate it, but it's just, that's it. You can't have a filled in recessive trait without a recessive genotype, okay? But let's keep going. What would that genotype be? Big A, little a. What's the next one? Same. How do you know that? Right? There are only essentially 2 possibilities for that cross. What are they? Huh? Oh, maybe there's only one. There's only one possibility. Big A, little a. So again, don't overcomplicate it. Don't try and think, well, like what's the probability of if these 2 and that one? Just do a Punnett square if you have to, right? Just do that and realize, oh, all 4 of them, that's the only option, right? That's the only way that it can turn out. Okay, what's this one? It's got to be little a, right? This is the recessive trait. It has to be homozygous recessive. What about the next 2? Can't tell, okay? Because technically, a big A, little a cross, what kind of genotypes can I get from that? I could get any of them, right? And so I definitely got that one there, but I could get either of those here. So if you see a pedigree with an A blank or an A dash, it's just saying we don't know yet. You'd have to look at their offspring. And since the pedigree doesn't go that far, it usually just gets denoted as A blank. So I just wanted to point that out so you guys don't go like in a mastering bio question and be like, why is there only one letter here? They only have one allele.
I don't understand why in the first and the parent it can't be heterozygous also, because we can still get 4 dominant offspring.
Who can't be heterozygous?
No, like the
this
mother,
one?
that's affected.
Yeah.
I just don't understand why it couldn't be heterozygous, because when you still get the same result in the offspring. You still have four unaffected children.
But there's no way for her to display the recessive trait if she's got a dominant allele. Yeah. So we overcomplicate that and we overthink it. It literally is as simple as she has to have the recessive genotype because she has the recessive trait. Does that answer that?
Yeah.
Okay. Other questions? Okay. So let's look at X-linked inheritance. This is X-linked recessive inheritance, and that's what we usually look at as far as X-linked traits. So first of all, what does it mean where the mother is a carrier? Okay, I'm assuming you guys said the right answer. One of her chromosomes has the recessive trait and one does not. So she's hetero or homozygous? Hetero. Right? She's something like that. Okay? The father, what is he? He's not really anything, right? And so that's why these usually actually get denoted in a different way. They usually get denoted like this so that you can then say that. So his X chromosome has the dominant trait. So he's not expressing because this is a recessive trait, right? And then the Y chromosome doesn't get factored in, right? It's too small to carry enough genes. And then the mother, one of the X chromosomes has the dominant, one of the X chromosomes has the recessive, okay? So these are the possible ways that they could share their gametes, right? So you could either end up with a female. See how those are two X chromosomes, two big chromosomes is a female. She's unaffected, and also it's important to note she's a non-carrier, right? Because both of her genes are dominant, okay? So she doesn't carry. You could also get a female who is a carrier, okay? Meaning one dominant, one recessive, just like the mother. You can get a male unaffected. You can't really have a male carrier, right? They can't carry because if they have it, right? And then you can get an affected male, okay? Meaning his X chromosome has it, and since he doesn't have another X chromosome to default to, another X chromosome to carry a dominant trait to suppress that recessive trait, not really any other option, he is going to show the trait, okay? One of the most common sex-linked recessive traits is color blindness, right? That's why you almost exclusively see colorblind males, right? In my session last year, I said that, and then a girl in the back raised her hand, and she's all, I'm colorblind. And I'm like, literally, in my 22 years, you are the first girl I've ever met who's colorblind. It's extremely rare, because she would have to have a father who is a carrier, or expresses, and a mother who's a carrier, so that she could end up with both, right? Which is far, far less likely, right? So I'm colorblind, if I haven't told you guys that yet. And what happened is my grandfather was colorblind, and my grandmother, we'll say that's empty, was unaffected non-carrier. So it actually ended up, my mom ended up a carrier, my dad is an unaffected male, and then, so it can kind of skip generations in that sense, because my uncle, their only son, ended up unaffected, right? So there's no like colorblind men will have colorblind sons, right? There's no guarantee that I will have colorblind sons or daughters. It's just more likely, right? He had 5 daughters and 1 son, so he also kind of skewed the curve a little bit there. Anyway, that's how X-linked recessive traits work. Does that make sense? We spent like 4 seconds on it in class, so expect 12 test questions. It's just how it works, guys. There will be test questions on it. There's not going to be there's never like a disproportionately large number of test questions on one thing, except maybe meiosis, but that again that's a pretty broad topic. So you'll have one or two maybe on something like this, but you've got to understand the mechanism, okay? Because they could be pretty deep questions. They could be pretty complex questions where you have to really understand the mechanics behind what's going on so you don't confuse yourself, right? Yeah.
I feel bad I walked in late, but how is it that your dad didn't have it but he still passed it on? Does that make sense?
So we'll draw it a different way.
Because if it's on the X chromosome, doesn't he only have one?
Yeah. So my father is unaffected, right? My mother is a carrier.
Oh, okay.
So I got my Y from him, but I got that X from her. I only have the affected X. And since I don't have another X chromosome to default to, does that help?
Yeah, so
Okay.
not just like none of your sisters could have had it because your dad didn't have it.
Right, impossible, because even if they got her unaffected X, they would still get her affected X, they would still get his unaffected X. So it is impossible that they could have been colorblind. They would be carriers, yeah. So if they ended up with that one and that one, they're carriers. If they ended up with that one and that one, they're non-carriers. And there's actually no way to know. Well, I guess you could get into their DNA, but that's like super expensive and there's no reason. There's no way to know until they have their own children, and then we know whether or not they're carriers. Fun fact for you, my wife's dad is colorblind, so we actually have a fairly higher chance of having colorblind girls if she's a carrier. So we'll find out someday. Or not. You never know. But that's a personal thing, and you guys don't. We've only been married for a few months, so let's not get carried away here. All right, just another way of looking at it, right? I just like to show multiple graphic images. Not graphic images, that's gross. I'm getting flustered, guys. I like to show multiple images in case, that last one didn't make sense for you. Maybe looking at it that way makes more sense, right? So this boy is me for color blindness, right? So he got the blue half, purple half, see? And the Y, and so he has no choice. It is forced upon him, and he will be shunned in elementary school for asking, what color is this crayon? I got yelled at by my 4th grade teacher for asking what color the Utah flag was, because I didn't know. Is it blue or purple? I couldn't tell the difference. She got really mad at me because she thought I was trying to be like a smart aleck. And so I cried, and she felt really bad. Because my mom called her, and she's like, my son came home crying. And she's like, well, he was trying to be a smart A. And she's like, well, he's colorblind, so. He really didn't know. And she was like, ugh. She was so nice to me for the rest of the year. And she was the mean teacher, too, so people were like, what did you do? And I was like, I have a disability. All right. Where were we? One more fun fact, because I care about colorblindness. Technical term is color vision deficiency syndrome, because I can see color, so I am not blind to color. I don't want to be that pretentious, so I don't say, I'm color vision deficient. People are, what? Yeah, I'm red-green. That's the most common kind of color blindness. It just means that like reds and greens that are kind of similar shades, or especially like greens and browns that are similar shades, just look the same. I don't have enough cones in my eyes, and so they just look similar. But I can see them. Although there are people with color vision deficiency syndrome that cannot see color at all. That's a different thing. 50 Shades of Gray. What is this? Let's get ourselves back on topic. I like talking about color blindness because I care. What is it? Nobody knows.
There are examples of different systems of like which sex chromosomes affect which gender?
Excellent. So in humans, we use the XY system, right? Because our chromosomes look like an X, and then the Y kind of looks like a Y. It just looks like a really squashed Y, I guess. I don't know whether or not chicken chromosomes look like Z's. That'd be pretty cool. That may just be what made the most sense because it follows, well, it starts before X and, right, W, X, Y, Z. I don't know the alphabet. Grasshoppers have an X0 system because their females have XX like we do, and their males just have an X, right? And then bees, honeybees, I don't know if all bees, have a haplodiploid system. So the females are diploid, the males are haploid, right? So I point this out only because you need to know that there are other sex chromosome systems, and just be familiar enough with them that if you saw like the phrase, you know, chickens have the ZW system, you don't go, ZW what? Like, I don't know what that means. It's talking about their sex chromosomes, right? You're not going to need to know how to do meiosis with a ZW cell. You're not going to need to know how to do meiosis with an X0 cell. I don't know how to do that. Just be familiar with these terms. That's about it. I don't know what else to tell you. This one's not super hard. So just call it good. Questions before I go on? I don't want to just assume people know what's happening. But you do, so I was right. All right, so how does our system work, right? We're very prideful. We really only care about how the XY system works. The XY system, this is mammals, right? It's not exclusive to humans, mammals. I don't know if all mammals, most mammals. The Y chromosome has a gene called SRY, just stands for sex determining region of Y. It acts as the master regulator. Does anybody remember what the heck a master regulator is?
Transcription factor?
It's a transcription factor. What's a transcription factor? Turns genes on and off. So if this were, per se, a master transcription factor, what would you guess that means? Think of it like a master switch or a master key. These are not plant cells, they're light switches. If this is the master regulator, okay, and it is connected to all of these, and we want to turn on these ones, what do we want to do? Do we want to go by individually and flip them all on, or do we want to just go to the master switch? So the SRY gene is the master switch. Everything related to the Y chromosome and therefore male characteristics are determined by the SRY gene. The SRY gene flips on if a Y chromosome is present and says, time to develop, male traits. Because the female sex is the default sex, right? We all start as basically a sexless fetus, well, embryo. And then the X chromosomes either, I don't want to say do nothing, but they just carry on as they are, and you end up female, unless the SRY gene on a Y chromosome turns on, then you differentiate into male, okay? So it's all the SRY gene. If there's not SRY gene present, you will not become male. This can go awry. There are cases where people have XXY chromosomes. I can't remember the medical term for this if anyone wants to Google that for me. Essentially, you are not hermaphroditic, but you have both sets of chromosomes, and so you have an SRY gene that turns on, but you still have a lot of female characteristics too. This is just errors in meiosis that accidentally put too many chromosomes in one cell. There are other ways that you can end up. You can get like 3 X chromosomes, which doesn't sound all that terrible, except that usually you end up dying in like your 20s. I can't remember why. Too many genes? I don't know. So there are ways that the XY system can go awry. Yeah. So mostly just understand how the SRY gene works as a master regulator and how that determines sex. Okay? Questions? X chromosome inactivation, something we also only spent 4 seconds on in class, but is important enough to reiterate so that you understand how it's working. First of all, what is X chromosome inactivation? Some brave soul. Raise a hand and help me out.
Is this the principle that he was talking about where there's an uneven balance of X chromosomes so that basically in females, one X chromosome will turn off, for lack of a better term, and allow it to be equal balance?
Yes. Because how many X chromosomes do males have? 1. But we get by okay, right? I mean, not great, but we're all right. You only need 1 X chromosome. But females have 2, which doesn't sound like an issue, but then you run the risk of twice as many issues, right? If you have twice as many chromosomes, you have twice as many things that can go wrong. So if you only need 1, just use 1. So essentially what happens is some proteins break this chromosome, doesn't matter which, it's a 50-50 chance, breaks it down and actually physically makes it smaller. And it's so small and so tightly packed, it can't be opened, therefore it cannot be copied, therefore it cannot express. So you literally just like smash the X chromosome down like a crushed can, and now it's useless, right? Because you just need the 1. Now this will do funky things in certain animals. It has 2 different fur color genes on its 2 different in its 2 different cells, right? And in one cell we happen to turn off the orange fur color, and in the other cell we happen to turn off the black fur color, but they both still express the other one, and you get this spotting. Aren't these called like turtle cats? Calico turtle. Maybe I'm thinking of like turtle ice cream that's got like caramel and chocolate. You guys know what I'm talking about. They're turtle cats. I like turtle cats better. We'll give him like a little. He's a turtle cat. He's a terrible turtle shell. So essentially the idea is that these X chromosomes, we have twice as many as we need. We turn off one of them, we inactivate one of them, and we express the other. Now this doesn't obviously occur in everyone, right? Otherwise every human woman would have like blonde and brown hair and a green eye and a blue eye and who knows what else, right? Like 1 foot that's way larger. No. So fortunately, no, unless you think that would be like really cool. But in certain species and with certain traits, you can see X inactivation. Now, what else does this remind you of? What else does this look like? Codominance, right? Because isn't codominance also, that's how we got like the white and roan spotted cow, the red and white spotted flower. So aren't we getting an orange and black spotted cat, basically? They are similar in phenotype, but they do not happen for the same reason. So don't take the comparison any further than they look similar, right? They do. And if we didn't know enough about genetics, maybe we would assume that this is codominance. Or maybe we would assume codominance is X inactivation, except that codominance happens in males and females. So part of the way that we first realized that this probably wasn't codominance is it only happened in females. And we went, codominance happens regardless of sex, but these cats, only females, end up calico turtle, right? So what's the difference then?
So codominance, that would mean that there's two dominant alleles and they both show up. But in this case, one of the smashed X chromosomes still displays the trait that it contains.
Perfect. So codominance, we're dealing with two dominant traits expressing at the same time. X chromosome inactivation, we're dealing with separate cells. Half of the X chromosomes have been inactivated, and it just so happens that the X chromosomes in separate cells that are still active are different. You can absolutely have X inactivation and never see it, right? If this cat only had black fur genes, would it undergo X inactivation? Yes. Every female mammal undergoes X inactivation in every single cell. You just wouldn't see it. You'd have a black cat, and unless you went and looked at the genes, you would just assume it was a black cat. But it very well could have orange genes, and every single one of them got turned off. Pretty low odds, but it could. Yeah, so this cat could be black, and you probably have seen this. If any of you guys have owned cats or know somebody who's owned cats, and they mate, a black cat and a black cat, and they get all these orange cats and half orange, half black cats, and like all kinds of cats, right? It's because that female had all kinds of genes, but they were all turned off, the ones that we weren't seeing at least, right? But all of a sudden there's orange babies, and you're like, did you cheat on Mr. Truffles? I don't know if cats have the concept of monogamy, but I don't think they do. Did you have a question?
Yeah. So like even if it's inactive, like an inactive X chromosome, it can still pass on those genes to its offspring.
Yes, that chromosome still exists and can still be passed on, and then could, through meiosis and through sexual reproduction and transfer and everything like that, then that chromosome could happen to be the one that isn't inactivated in an offspring.
But the crushing doesn't change the genes or how they would like show up in the kids?
Nope, just keeps them from expressing. It just smushes it down, and it's impossible to open that gene up and get genetic information out of it, but it can still pass on.
So why don't female humans look like that?
Say what?
Why don't human females look like that?
That I do not know. My best guess would be we don't have any kind of traits that follow X inactivation. I'm not really 100% on why humans don't do the same thing.
Is it decided randomly which X chromosome will become inactive?
Yep, 50-50 chance, same as independent assortment, right? Nobody's over there saying, let's do this one, let's do that one. It's just
For every cell.
for every single cell, whichever one. So there's a 50-50 chance on that top one, independent 50-50 chance on that bottom one. They just end up how they end up.
When does X inactivation occur?
That is an excellent question. Did he mention that in class? Does anybody know? When does X inactivation occur? That makes sense to me. It would be early in fetal development. It's going to obviously be after sexual reproduction and after some of that initial meiosis. They have to get passed on first. Not totally positive on the time frame. Although if it wasn't mentioned in class, you won't see a test question on it. Not to say that you shouldn't desire learning, but not going to see a test question on it. I thought I saw another hand, yeah.
Just kind of a random question. Did this ever happen in a male X chromosome inactivation?
I don't know. I would hesitate to say never, because there's always some kind of exception, and somebody Googles it and they're like, well, you're wrong. And I'm like, all right. Not that I know of. I don't see a reason that it couldn't if something went wrong. but it's not supposed to. So in a normal cell, it wouldn't do it, because it would go, oh, we're just expressing one gene, don't mess with it. But yeah, I mean, if some proteins got screwed up, maybe. You'd probably die, though, because you would have no genes. So it's like, you'd stop at your toes and nothing else happens. That's not how fetuses develop, but you know what I'm saying.
So how often does this actually happen?
How do you mean? Like, how often does X chromosome inactivation happen? Always.
Okay.
Every cell in every female mammal is supposed to. Again, there's always that like weird exception, but you should always get half of your X chromosomes turned off in every cell. Always. Other questions? So that's the end of my slides. What I wanted to show you guys before you decide to already bail out. I know as soon as PowerPoint's closed, people are like, we're done. Don't run off. I'm going to torture you. I still have, 15 minutes? How many of you guys saw the announcement that we're not going to be announcing our slides anymore? We're not posting announcements with our slides in them anymore. We got some strongly worded feedback on the, what do you call them, midterm evaluations, of people saying, I'm tired of getting 10 emails a day. First, I'd like to point out, it's like 3 or 4, so calm down. But also, I'd also like to point out, welcome to being an adult. But we also get where you're coming from, because we announce that we're having a session, and then the same day we announce, here are some slides. And so what we've done instead, come into your Canvas page, right? Everybody's peeping my classes. Go to just the home page of Bio 1610. Go to the SI and UTF information, and then Hillary has created essentially a blog, but we're not going to tell you what we did over the weekend because that's none of your business. If you would like me to, I will tell you, but it's usually studying and Walking Dead, so no longer at the same time. You would just click, so if you wanted to look at, this is from last week, but if you wanted my slides from Monday's session, you just click on it and it automatically downloads, right? So it's the same as an announcement, but now you don't get an email every time we do it. So if you start wondering where the heck did all of the PowerPoints go, they're just now in this blog instead. Captions. I know. That's one of my favorite things. I sit there during class. I'm not going to lie to you guys. If you ever look back at me, I'm on like the, what is that, the north, pretty much like the north corner of the classroom. If you look back at me, I'm just grinning like while I try and think of funny announcement titles to come up with. I've had some good ones. I've had some less than good ones. I haven't done my sexiest enzyme one. I'm thinking about it. Did I tell you guys about that? I got in trouble. I didn't get in trouble. One person was like, you need to stop with all the sexual comments. And I was like, I made one genetics joke about unzipping genes. Because you do. Yeah, so we'll see. End of the semester, when no one can get mad at me. Well, we still have end of semester evaluations. I want to show you guys something else. Because I've had so many people emailing me lately. I don't want that to sound ungrateful. I love getting emails from you guys. It's what I'm here for. It's part of my job. But I've had a lot of people coming to me and saying, I'm not doing as well in the class as I would like to, and the SIs help, but I definitely feel like I need more help. A lot of people ask me to do like one-on-one tutoring with them. As an SI, I made like a contractual agreement not to do any kind of outside tutoring. I would literally get fired if I did. I've thought about it, though. I would make some killer money, more than they pay me right now. The Academic Success Center is usually what I refer people to, right? So you just go to the my.usu.edu page. You just search Academic Success Center. It's the first one that comes up. You click on it. It brings you to here. There are tutoring links, right? So if you want to get some help with tutoring, you can go to the tutoring schedule, tutoring advisor, and find help in math, biology, physics. They tutor all kinds of stuff, right? So obviously not just for this class. There's the link to the SI, so you can get some of our stuff. There's test taking prep, assessment, strategies, stress management stuff, study helps, right? Assessments, all kinds of other resources that you can use. There are so many extra things that can go beyond this class. So if you really feel like you need more help in it, then this is one way to do it. We got about 10 minutes left. If you guys want, we can just ask questions about anything for exam 3. We can practice Punnett squares and pedigrees. You can leave. You want to practice? What do you want to practice? What?
Hybrid crosses and,
Dihybrid crosses?
yeah.
All right.
You know, it's a faster way to find the genotype of all the potential genotypes in that hybrid cross, or you just have to actually count them all up?
In this class, you essentially just have to count them all up. Fortunately, we don't have you do a quad hybrid cross or anything insane with 200 possibilities. You'll probably have max 16. And so if you can do Punnett squares relatively quickly, I will teach you. Thank you. Did anybody not get the roll? Okay. Oh, back there. You'll just have to come up front then. There is one trick I'll teach you. If you get one of these, right, how would you make that Punnett square? Somebody walk me through it. Here's some mumbling. You're making hand gestures. Tell me what to do. You, yeah,
Me?
right there.
Oh, I just set up the Punnett square so it would be two big A's on top and then the big A on the side, and you just mash them. So.
Right, and then you just pull stuff down and pull stuff in, right?
Yeah.
So watch this. Big A, big A, big A, big A, big A, little a, big A, little a, right? So what is the genotypic ratio here? Right? Phenotypic ratio.
1.
Assuming big A means dominant, it's just 1, right? So what did this row even give me? Nothing. If you have a homozygote, you can just ignore its second one, right? Because regardless, you're just going to get the same thing every time, right? So if you have A, B, B with an A, B, B, instead of going through the painstaking process of saying, well, we got big A, big B, big A, big B. Am I doing that right? Big A, big B, big A, big B. And then a big A, big B, big A, little b, big A, or no. Little a, big B, little b. This one's going to do the same thing every single time, right? So just ignore that one and just do that. Your ratio is not going to change because it's a ratio. Again, it is safer to draw the whole Punnett square, but if you practice that enough times that you're confident in what you're doing, that you know that when you sit down in a test and you draw a Punnett square, I can ignore that second bit of the heterozygote, then that's okay, right? Go ahead and do that. But practice it first, right? Don't just be like, yeah, Parker said something about cross that one out. You might screw up some of your numbers if you don't know which ones you're crossing out and where. But does everyone see what I did and why? I just discounted that second bit from the homozygote, because it's homozygous. So you're getting the same stuff from each of them, okay? If you're not comfortable with that, either come up and talk to me and we'll take a second to work on it, or just don't do it. Much safer, right? I don't want to take the time to make a pedigree because I feel like that will take all of our time. So let's just Google. This is literally what I do, guys. Don't think I'm some kind of super, it's all dog food. That S makes a difference. Let's see. We'll use this one. I'm just making these on the fly. I came so prepared. Help me label these with their genotypes. So, first, actually, help me decide dominant or recessive. Oh, I'm getting some confusion here. If it were recessive, what would I need to see? Do you guys see any unaffected parents having affected children? I don't either. Again, you guys have to realize I just Googled this, so I don't see any. I would say this is dominant, personally, because I don't see any unaffected parents having affected children. So I'm going to say with a fair degree of confidence, we're looking at a dominant trait here, okay? So don't just look at this and go, filled in means dominant. It does not mean dominant. Do not do that, please. Someone will do it on the test. I promise you. Someone will be like, oh, it's dominant. It's got filled in circles. Have you ever seen a pedigree with absolutely no filled in circles? Like, you don't. Okay, so I guess for funsies, autosomal or X-linked? Autosomal. How do you know that? It's pretty much an even split between male and female, right? Even if it's a little bit more male, which I don't think it is. Yeah, it's actually more female in this one, isn't it? Again, if they're close, in order to say this is definitely sex-linked, you got to see like pretty much only males, maybe 5 or 10% females expressing it, right? That's the only way you can really confidently say this is sex-linked. So we don't see that, so it's got to be autosomal. Okay, so what's this phenotype?
It's recessive.
Somebody says homozygous recessive, so we'll use A's. So you're saying it's little a, little a? Okay. Does anybody disagree?
I disagree because you said because they agreed that it was all dominant.
It's a dominant trait, right? Red meaning affected, so it has to be A, and we'll leave that blank for a second, right? Because technically we don't know yet if that's a little a or a big A right now, okay? This one, what is his genotype? This one's got to be homozygous recessive, right? Because it's a dominant trait, but he's not displaying. So the only way he could not display a dominant trait is if he is homozygous recessive, right? Because if he's homozygous dominant, he's going to show it. But if he's heterozygous, he's still got that big A. He's still going to display the trait. So do you guys see why I kind of refer to these as logic puzzles? You got to logic it out. You got to think this through and go, okay, it's a dominant trait. He's not showing it. He's got to be homozygous recessive. Okay. So let's say, what's this one? What's his genotype? It's definitely got a big A. So do you think he has another big A or a little a? It's got to be little a, right? Because where did he get the big A from? Got the big A from mom. And the only other thing he can get then is a little a, right? Again, logic puzzle. Think it through. Say to yourself, he got the big A from mom. What could he get from dad? All right, so again, same story. What's here? Big A, little a, right? Same options. What's here? It's got to be little a, because they're not expressing, right? If they're not expressing and it's a dominant trait, you are 100% certain that they are homozygous recessive, right? So I'm not even going to take the time to do every single one of those. Just know the blue ones are little a, little a. Okay, so what's this guy going to be? All right. What's this guy going to be? Huh? It's got to be big A, little a, right? He got big A from dad. But his only other option is little a then from mom. Yeah?
So that means that the first, like the top affected red ones, that has to be a little a as the second one, or else they can have double recessive children, right?
So you're saying this one definitely has to be big A, a big A, little a, because why?
Because if it was big A, then it couldn't have double recessive children.
Saying it has to be that, otherwise there's no way to get that. Yep, that's exactly right. So the only way for him to get little a and little a is to get little a from dad, but he has to be getting a little a from mom too, right? Otherwise, if mom had been big A, then all of these would be big A, little a. So that's a very good catch. Again, what's this one going to be? Same story, big A, little a, right? Getting a big A from dad, little a from mom. And then that one, big A, little a, right? By the same logic. Big A from mom, little a from dad. And then obviously all of the blue ones we know are going to be little a, little a.
So does that mean the wild type phenotype for this would be the recessive homozygous?
The wild type for this,
Because it's the most common? Or is that playing in a different way?
the wild type, because the wild type just means the most common one, right? Yeah, so it looks like we got a pretty strong majority for homozygous recessive, so that would be your wild type, yeah. All right, that's our time. Thank you for being here. Keep practicing those. Literally just do what I did. Go and Google them. Yeah, and practice that. Practice Punnett squares. Use that Punnett square calculator.